Yes. All right. Good morning or good afternoon, everybody. Welcome to the 5th edition of the BioREPS Online Seminar. I'll be your chair today, and my name is Devika Narain. I'm a lecturer researcher at Van Gaal University Applied Sciences, and we will have 4 amazing presenters today. So we will start with Professor Max Huisden, and he will tell us something about environmental perspective on bioactive molecules. He will have 30 minutes available, and after that we will move to the next speaker. So Professor Max, you can take the floor.
Okay, thank you, Devika, and a good morning and a good afternoon to all of you. Can you hear me okay before I continue?
Yes.
Yeah, so environmental sciences is a very broad perspective. It's a very broad topic because we can divide it into the macro environment and the micro environment. The macro environment consisting of the 3 big compartments, air, water, and soil. And the microenvironment is actually what's happening inside the living organisms. So within this context of biology and BioREPS, it is very honoring to me to give you a perspective on the environmental aspects of this topic. I'll dive deep, so I'll go on a molecular level. Putting in perspective, bioactive molecules. So when we're talking environment and bioactive molecules, it is actually good to know that bioactive molecules are chemical compounds that exist inside nature, and they actually have an effect on a physiological level and on a genetic level of living organisms. So a lot of times people think that the genetics of an organism are basically determining the phenotypical expression. But currently there is a scientific field that shows that environmental impact is of the utmost importance through processes that are called epigenetics, where a process that is called methylation plays an important role. So methylation, I promise to go deep, so we'll dive right into the cell. Methylation is actually a process that takes place in the nucleus of the cell, where the nuclear DNA is present. And as you know, the DNA is wrapped around histones. Histones are proteins inside the cell that actually determine the distance from genes that can be regulated. And this distance is actually important because it actually expresses, or it allows genes to be regulated. Upregulation and downregulation are important topics when we're talking about especially protein synthesis within the cell. So currently it is known that the protein synthesis is basically orchestrated by environmental molecules, mostly molecules that can be in the macro environment and in the micro environment, like I presented before. So I like to keep this interactive. So if there are any questions, and especially if some of the cameras could be on, that would be wonderful, because currently I'm talking to myself, which can be, I don't want to nod off in the middle of the presentation. So thanks for turning back on. But in any case, and if there are any questions during the presentation, I like to be interactive, so feel free to jump in. So from my perspective, this topic of epigenetics is leading in my research and in my academic endeavors. So now I'll talk a little bit about what we do. I'm chairman of the Department of Environmental Sciences. It has been for the past 14, maybe 16 years. And my background is actually in pharmaceutical science, in toxicology, and in forensic science. So I'm very much interested in reconstruction research when it comes to molecules that cause like an physiological happening, like an event in, it can be a calamity or it can be a crime or anything like that. So the laboratory that we developed at the University of Suriname is a laboratory that is certified, and we have equipped ourselves to do research within this area. So what we have now present is an HPLC, which is high pressure liquid chromatography, which gives us the opportunity to measure chemical molecules quantitatively and qualitatively. We have an qPCR, which is quantitative real-time PCR, which gives us the opportunity to look at the genetic level to see what the impact is on the physiology of organisms as it comes to things that happen in the environment, but also for reconstruction. Like I mentioned, forensic science is one of the things that we do. We have a 96 well plate reader, which is a spectrophotometry that gives us an opportunity to look at interactions of chemical compounds and how that interaction proceeds real time also. And we have a mercury analyzer that is actually focused on metal mercury. Metal mercury is the most toxic form of mercury. And as you guys know, mercury is like a hot topic in Suriname because of the gold rush. It is being used in artisanal gold scale mining, where lots of mercury, too much mercury is getting into the environment. Mostly because of the ineffective use of this heavy metal. Normally, you would get maybe a one-on-one, like 1 kilogram of gold using 1 kilogram of mercury to extract, and then you could potentially recycle the mercury in such a way that it doesn't get into the environment. But the people that work in this area, which are small scale gold miners, mostly uneducated people that are trying to make a lot of money quickly, they can use up to 3 times or even 5 times the amount of mercury, and they do not recycle to a satisfactory level. So a lot of mercury is getting into the environment. So mercury enters the atmospheric cycle because mercury is very interesting chemical. It evaporates at room temperature. It gets into the atmosphere and it can be there for many months, even years. And you understand what happens. It is taken away with the wind and can come back on Earth when it gets into a polar form. So it becomes water soluble and gets back on Earth with the precipitation, where it is actually converted by microorganisms to metal mercury, organic mercury. And this organic mercury is entering the fish cycle. You know, small fish eat plankton and small plants, and it's those small plants that actually convert elemental mercury into organic mercury. So it bioaccumulates in the bodies of the small fish, and they are being eaten by other fish. So it also biomagnifies in the fish cycle. And people tend to love the biggest fish, and those are mostly the predatory fish, which exposes us to a lot of mercury potentially, not only where they mine for gold, but also in other areas such as Paramaribo and surrounding areas where we're being exposed by eating fish that is contaminated with metal. So in our laboratory, we focus on metal mercury organic form. It's a neurotoxin, and it is a teratogen, meaning that it's the system negatively. but it's also a teratogen, so it goes over to the next generations, where it can cause developmental issues, especially cognitive issues. And so that's the mercury part. Interesting to say, because we have ratified about 8 years ago the Minamata Convention in Suriname, where we need to actually outphase the use of mercury. But in the process, people are very inventive. So they have come with other chemical compounds, mostly engineered products that are being used as a substitute for mercury right now. But some of those contain cyanide. And as you guys know, cyanide is way more toxic than mercury in a different way. Where mercury is a neurotoxin and a teratogen, but it actually causes chronic intoxication. Cyanide is a acute toxin, and very small amounts, it can be lethal to living organisms and humans, especially. So when that has become a problem in Suriname from an environmental perspective, but it threatens our biodiversity at a large scale. So this is why I emphasize this within this meeting, because I know that the topic is mostly focused toward biology, but from an environmental standpoint, on a molecular level, where toxicology and forensic science and my field of research kind of highlights the importance of molecular level, micro environmental level interaction with biodiversity and life in general. So let's see, how many minutes has it been?
13 minutes. So Max, the bioaccumulation is, do you know of any studies on the fish, whether some species pick up the mercury or the cyanide more quickly than others, and does it end up in people then?
Yes, definitely. So to answer your question, a lot of studies have been done by local and international scientists, focusing mostly on the areas in the interior where gold mining is an issue and where mercury gets into the environment. And most of those studies have looked at the accumulation in fish species. So this is where, with my background, I saw a niche in the market, and I started focusing on metal. Sorry. Excuse me. Yeah, excuse me. I started focusing on metal mercury, and this is why we have this specialized equipment in the laboratory. And that led us to do some additional research, and we have actually published in peer-reviewed journals the exposure of people in Suriname, especially in the coastal area, Paramaribo and surrounding areas, where we looked at the exposure through fish consumption, looking at two big sources, like fish that comes from the fresh market, which would be unprocessed fish that is freshly sold. We looked at 4 big markets, did a study to see which fish are being sold mostly, so the most popular fish, so to speak. And we looked at processed fish in about 50 supermarkets, and those were all like randomly chosen. And looking at the frozen fish section, where fish is being processed, we got an idea of the fish that are being sold most prominently. So the most popular frozen fish products. And from those top 3 in each section, we did an analysis to see whether the levels of mercury accumulation is in the bodies of those fish. Other than that, we did a large-scale survey of the Surinamese households to see what their habits are, their nutritional habits. How often do they eat these fish, and what is the serving size? What is the consistency of the family? Like things like an age, weight, body weight, etc. And so we made an extrapolation, a correlation and an extrapolation to see how much mercury actually accumulates in Surinamese people in that eat fish. And it was alarming to see that in some fish species, mostly in predatory fish, such as the piranha, are contributing significantly to the exposure of mercury. But most concerning is actually that the indigenous people in Suriname, they don't eat fish like 2 times a week or 3 times a week. They actually eat fish 2 to 3 times a day. So their level of exposure is way higher than the average Surinamese citizen. So I hope I answered your question, but we had some interesting research done in Suriname, and most of it is published, peer-reviewed, so internationally available.
Yeah, great. Thank you.
Are there any more questions?
Yes, so now we've moved on to questions and in the interactive part. So please raise your hand or just ask your question.
If I can elaborate on anything that I've said, I'd be happy to. So feel free to ask.
If I may ask a question, Professor Huisden, thank you for your talk. And I was wondering what bioactive molecules have the focus for the next coming years. So what do you think should be focused in on or research even more or deeper regarding environmental and human health?
Yeah, thank you for that question, Devika, because that is actually the reason that my research is staying focused in this area, bioactive molecules. I actually have a Lehrstuhl at the moment in environmental sciences, but this Lehrstuhl is bioactive molecules, how they affect health, and they can be used to boost the economy of Suriname in a sustainable green way fashion. In contrary to mining activities that maybe lead to quick cash, but are detrimental to health and environment. So to answer your question, my research is focused on bioactive molecules within the area of non-timber forest products. So I think that should be it should become more prominent in the coming months and years, because it gives the opportunity to look at bioactive molecules in plant species and how they can actually sustain not only, but how they can restore health. And Suriname is the greenest country on earth, I'm proud to say. We have 93% of forest coverage, and within this vast forest, we have a lot of those bioactive molecules that are actually anecdotally already used in for health purposes. Not only, but Suriname has a very rich culture. So we have people from all over the world that came together here in Suriname, and we took with us the anecdotal evidence from our ancestors from all over the world. So a lot of knowledge from how we call them osodresis, how these osodresis, how these plants and herbs and things that we actually use on a regular basis as a people, how they can affect health. But it is all anecdotal evidence. So it goes from father to son and etc. So that information may be helpful, but it's not scientifically researched And my research in bioactive molecules has a wide range. It ranges from environmental issues such as mercury, cyanide, and other industrial toxins such as maybe pesticides and herbicides, etc., how they can be detrimental to health. But on the other side of the spectrum, we look at validation of bioactive molecules in order to sustain and restore health. So we actually develop and help design medicine from nature, validate it in order to potentially make it green, a green a green pillar,
Yes, a green
a
pillar.
green economy. You know, it's sustainable. It's a non-timber forest products. There is internationally a huge market for it because the pharmaceutical industry is mostly focused on the treatment of symptoms, but the underlying conditions that cause the health issues are mostly overlooked. But within this area of naturopathic medicine, and my PhD is in pharmaceutical science, my focus has been on the disease of Parkinson's disease, and the research that I've done. And just just a couple of weeks ago, one of our students actually, I handed over the baton of my research in bioactive molecules.
Nice.
Devika, you probably know Leandro.
Yes.
Leandro
Yeah.
So, I'll just use 30 seconds to explain this because it's very interesting. So Leandro came into environmental sciences as a first-year student, but he was trying to get into medical school to become a medical doctor. But when he followed my classes, and my background is in naturopathic medicine and pharmaceutical science, you know, and the microenvironment, like I explained before, is very prominent in my courses that I teach at the university. So he became actually interested in environmental sciences from a medical perspective. So he pursued his environmental studies, even though he wanted to do medical school. But then we reached the point where Leandro had to graduate. And I told him, you know, I'm busy with the lab. HPLC is coming. I'm writing grants. It's coming. Just hold on. In the meantime, he put his environmental sciences studies on hold, waiting for me. I'm sorry to say. And he went to medical school. He finished his medical degree and came back to finish his environmental sciences degree, looking at a plant product, Mucuna pruriens, which is a legume with high alpha content. And he actually looked at coenzyme Q and L-DOPA in Mucuna pruriens under Surinamese conditions. And he graduated like 2 weeks ago. Actually, last week I handed him his diploma. So now Leandro degree. He's a medical doctor. He's currently still an assistant, but in a year he'll become like a full-fledged medical doctor. And he has his environmental degree in Mucuna pruriens with the focus on bioactive molecules, like I'm interested in. So that was interesting. But I hope I answered your question, Devika.
Yes, you did. Thank you very much. And it's nice to see that, Leandro has grown and that you're working together. So maybe we have room for one more question, and we can move on to the next speaker.
Anybody? Because otherwise I have a question. Yeah, so to bring the plants and the fish maybe together, because you said there is bioaccumulation of the metals through the fish, but I guess in agriculture you may also get accumulation in the plants. Has that been studied as well? Because going through the air on plants, or maybe even in the fruits as non-timber forest products, it may also accumulate in fruits.
Yeah, so that question is very interesting because it takes us back to the very essence of this topic. Because when we're talking about bioactive molecules produced by plants, we need to be aware that these plants produce those molecules based on their environment and on their need. So things like the elevation or the topography, or even things like the time of the month, the lunar position, the intensity of the sun during the day, things like the soil consistency, everything in the environment affects the plants and how they produce at a molecular level these proteins. Because those bioactive molecules, they are proteins that are being produced by the plant for a purpose. So if you put a plant in an environment that is polluted, it will affect the proteins that are being produced. So to answer your question, there is a very strong link. There is a very strong link between environmental factors, the growth and the uptake of contaminants by the root system and how it is expressed in the vegetative parts of the plant. And of course, non-timber forest products within my perspective, like in natural medicine, not only, but also in nutrition. So when vegetables, fruits, etc., can actually express the contaminations with heavy metals or other chemical compounds that are in the environment. Did I answer your question, Hans?
Yeah, so I think directly the metals can end up in plants, which then can be eaten by people or animals, but also the metals will affect maybe the presence or nature of the bioactive molecules. Is that correct?
Yeah, definitely, but especially in the context of Suriname, because as you guys know, Suriname is a very fertile country, and especially our coastal area consists of clay, heavy clay, that has been deposited throughout the centuries from the Amazon. And this clay is very fertile because clay has the ability to bind minerals and metals, etc. It just binds them. And when plants are present within this clay and water is present, these compounds actually become available. So the root system transports them into the vegetative parts of the plant. And that goes for plant nutrition, but it also goes for like contaminants, heavy metals, and things like that, which are being expressed in the plants. Yes.
Cool. Thanks.